Hi, do you know me? I'm George Heufelder. Some of you may know me as that old guy who gets up at MHOA meetings every year and gives a talk and ruins your dessert by talking about layer cakes and septic systems and mixing the two right after lunch. But today I'd like to talk to you about an exciting project that we're starting here at MassTech in cooperation with DEP and a working group, a Title V working group, that got together to answer the question, how much is too much or how much is too little? And the project involves a very important subject. Wait for it. Viruses. If you're a health agent, you're probably tired of hearing about it. But long before COVID and a galaxy far away, if you can even remember before COVID, there was a group of folks who got together to try to decide whether the distance to groundwater for Title V systems was adequate enough or whether it was too adequate, whether it was too restrictive. So along with a bunch of rascals at DEP and the working group, they put together a study with us folks here at MassTech, Massachusetts Alternative Septic System Test Center, in order to sort of get at the issue. It involved a large endeavor to put together a laboratory that could actually culture certain viruses and to put together the infrastructure to actually test it. And it takes a team of people. You've heard it takes a village, it takes a team. It takes a large team to start putting together both the infrastructure of the laboratory as well as the field itself to actually test these things. So the viruses, what viruses would we use? And today we'd start to think about using viruses that aren't harmful. And in your body, if you have ever eaten a pepper plant, are some phage viruses, as well as many other phage viruses. These are viruses that attack bacteria. And when they attack bacteria, they do one thing. Like viruses in the real world, they just multiply. And so they're in wastewater, and since they move very similar to real viruses or human pathogenic viruses, then we can use them as sort of a surrogate measure, much like we use fecal coliform in bathing beaches or E. coli or enterococci, except we needed something very small, something small enough and moves as similarly as a human virus. So, along with a vital member of the team, Dr. Pancorbo up at the Wall Experiment Station, we worked to see what viruses we could use that would be easily culturable, would not be dangerous, but that we could concurrently take samples and run up to the Wall Experiment Station and have run for real viruses. So that's the project, and I'm just going to briefly describe the infrastructure that actually tests the theories about how much groundwater, how much separation we need to groundwater. And that is that we have constructed a number of different scenarios that are very similar to Title V scenarios. The one which we're all familiar with, 5 feet to groundwater, 4 feet to groundwater, 3 feet to groundwater, and 2 feet to groundwater. So we have built 5 replicates of each of those treatments under gravity distribution, where there's 5 foot of separation, 4 foot, 3 foot, and 2 foot separation. In addition to that, we know that there's another mode of distribution on top of a leach field or into a leach field at the soil interface, and that is low pressure distribution. So for those scenarios, we tried 4 feet, 3 feet, and 2 feet. So with 5 replicates, count them 5, I think I still have 5, 5 replicates, 7 different treatments. There are 35 cells out there that are actually going to be used to test this out. Now you can imagine that that's going to take quite a feat to get 35 cells with the exact same dosage, timed the same so that all the variables can be fixed or similar in each of those situations so we can compare. So what we will be doing is, Brian Baumgartel, who has been working on the wiring and the sort of dosing structure there, has figured out how to get these doses precisely measured to each of these systems. So I'm going to let that go until we actually can walk around the site or show the site a little bit better. I know we have some good shots of it, so you can better visualize it. And I'll be back at the end as we introduce the team and what their different duties are in this particular project. So I'll return to talk about some of the metadata. Metadata is what we already know at the beginning of the project, because we do know some things at the beginning. We've done some virus work here before, and we just have not done it in such a comprehensive way. And since that's what's needed in this project, you'll see that this is where we're headed with it. By the end of this project, we will know how human viruses move beneath the septic system, simply because we'll have the surrogate measures paired up with the measures that Dr. Pancorbo will be making up at Wall Experiment Station.
I'm Kathy Regan. I'm the microbiologist here at the Mass TC, and I work on a project called the Virus Project. To begin the Virus Project, we have to start with something before the virus, which is E. coli. E. coli is a bacterium that lives in the guts of all warm-blooded animals, including humans. And the reason it's of interest to us is that because it lives in our gut, it's expelled in our feces or our poop. It has long been used as an indicator of water contamination, drinking water, recreational water, lots of different waters. Lots of people are familiar with the idea of the coliform count at a beach in the summertime. Don't go swimming if the coliform count is too high. Unfortunately, although it's a good indicator of bacterial contamination, it is a less good indicator of viral contamination in water. Viruses can diffuse further over greater distances than bacteria can. So if we're interested in viral contamination of water or wastewater, we are better off using a virus to track it rather than a bacterium. The viruses of bacteria are called bacteriophages, and those that are particular to E. coli are called coliphages or phages. In our study, we're going to be using 2 phages of E. coli, one for each of 2 specific strains of E. coli that we're looking at. The other advantage to using viruses or phages as indicators of pollution is that they're much more resistant to disinfection. So even if we can wipe out the bacteria in a water stream or a wastewater stream, we can't always get rid of the virus. Of course, under current coronavirus situation, the presence of virus in our wastewater has become of much greater interest to us, and the need to understand how viruses move in the water stream has become of much more interest to us. So viruses and phages are really beautiful, actually. They look like beautiful aliens. A lot of them have heads, legs, and tails, including our phages. Coronavirus does not have a legs and a tail. It only has a head, but they have different mechanisms by which they enter their host. But the point is they have a host, and when they enter the host, they eat the host. So we use the phages of our E. coli to eat our E. coli hosts, and in the process of eating those hosts, we can detect their presence, and we can also detect their concentrations. And we can use phages as surrogates or stand-ins for more pathogenic viruses because they only want to eat bacteria. They don't want to eat humans. So they're very safe to handle, very easy to handle. The virus project here at Mass TC is using a set of test septic systems, 4 different soil depths and 2 different delivery systems of one wastewater stream to follow the movement of viruses, in this case our phages, through the soil absorption systems and try and learn the rate at which they are slowed or taken up by the soil as they move through the soil depth. And concurrent with the phage analyses that we're doing, we're also going to be measuring 3 different coliform bacteria that are present in wastewater so that we can relate the bacterial load in the samples that we take with the viral load that we detect in our process. So the whole process of doing this analysis is basically a 3-day process, and it involves diluting the influent sample, which is going to be too high in phage for us to be able to count, and concentrating the samples that we take at the different soil depths, because as the virus gets held back in the soil, it's going to be less and less concentrated. So we have to have a way of still being able to see it. But the 3-day process involves, on day 1, giving our 2 E. coli bacteria each their food in a tube and their own antibiotic. And the antibiotic is such that only the bacterium that we want is going to grow in that tube. And that grows overnight. And what happens is that the cells grow and then they reach a steady state. So they've acclimated to the food we've given them and the antibiotic that we've given them, and they're really ready, happy. On day 2 in the morning, we transfer some of that material to a fresh food supply and antibiotic supply. And now that these cells have gotten very comfortable in this environment, they start to grow really, really quickly. And what we want is to use them at that point at which they're growing most quickly, because that's when they're going to be most vulnerable to any phage that's present in the sample when we prepare the sample. So after the mid-log, what's called mid-log phase, when those are ready, we then have another batch of food, antibiotics, and sample, which is either diluted if it's the influent, or concentrated, which I'll explain in a minute, if it's the sample from depth. Okay, the filtration process for concentrating samples is pretty interesting. We use dialysis filters, actually, which have very, very, very fine capture potential. And what we do is we start out with 2 liters or 2,000 milliliters of sample, and we pump it through the filter and hope that everything that is in the sample gets trapped on the filter. But then how do you get it off of the filter? After you've run all 2,000 mils through the filter, we have a special little solution that we use, and we hook the pump up so that it runs in a circle. And we run it back and forth in the circle, clockwise and counterclockwise. And what that does is it flushes all of the trapped material that was in the filter and captures it in this elution solution, which we then divide up and just treat as though it's a normal sample. So each of the host bacteria gets 100 mils of the concentrated, eluted sample. And we mix them all together and pour them into Petri dishes, plates. So we pour them into plates, we put them into the incubator at 37 degrees C, which is body temperature, human body temperature. It's exactly what E. coli wants to grow in and live in. And if there's phage present in the samples, they grow too, and they grow by eating the bacteria that are present in the plate. So on day 3, when we take the plates out of the incubator, if we had phage in those samples, we will have holes in the little holes that we can count on the plate, because they will have eaten into the bacteria that grew on the surface of the plate. So the bacteria that grow on the surface of the plate is called a bacterial lawn, and the little holes that the phage make when they eat the bacteria are called plaques. So we count the number of plaque forming units, and we relate that back to the volume of sample or the diluted volume of influent that we use to put into these test septic system cells. We've done some preliminary tests, and what we've learned is that the influent, the source water that we're using, the source wastewater, is very high in both phages, such that we have to dilute it by a factor of 10 in order to have few enough holes or plaques on our plate to be countable. So the incoming waste stream we know has a lot of these phages present. And so we dilute the sample in order to be able to count it, and then we just correct for that dilution at the end. As the sample goes through the soil absorption system, however, the concentration gets lower, and those are the samples that we run through a dialysis filter in order to concentrate on the filter, such that we then can also do another calculation to relate what we've captured in, say, 100 mils of sample back to the sometimes 2,000 mils of sample that we have to originally run through the filter in order to capture enough phage. The method that we're using is pretty well established by a number of EPA methods, 1602, 1642, and 1643. And those methods have laid out for us the requirements that we demonstrate the proficiency to actually capture and measure the quantities of phage that we're interested in. So before we could even start the experiment, we had to demonstrate that we were able to, using the methods that they lay out, find or record, detect the concentration of each of the two phages that we're using, which we did. So now we're ready to rock and roll.
So for the virus project, it's really important that all of our staff are on the same base, and so we need to communicate that information. So a lot of my role will be related to quality assurance. So here at MassTC, we do have a fairly robust process when it comes to documentation. Documents have to be controlled, so they are thoroughly researched and reviewed, and then the director of MassTC, Brian Baumgartel, has to give the okay on them, at which point they're distributed to all staff and they have to be used by all staff. So when it comes to this project, it's going to be important that we make sure we're all following the same method and that we can duplicate our efforts.
So if you can imagine, just a few short months ago, behind me, where you see a trailer in the background, was a large hole akin to what an excavator could do just by going crazy for a couple of days. And our challenge at this point was actually to construct 35 cells in a very small area. Small area because we really needed to keep the feeds for those 35 cells very short. So the residence time in the pipe was short, and it would simulate a Title V system. So we had a little bit of a challenge. How can you get down deep enough to underdrain a 5-foot deep leach field, still have enough freeboard to collect a sample in a 2-liter bottle, and also to allow for some drainage, so you have to create the drainage. So we chose to use 5,000-gallon tank shims. The tank shim is actually the portion of the tank that is bottomless and topless, and they usually use those shims to increase the volume inside a tank. So we had them custom made to different depths so that we could actually work down there and fill the cells little by little without the danger of them caving in on us, because that would definitely ruin your day. So one of them goes down as deep as about 8.5 feet. That's a 5-foot cell. Has a drainage area with some slope into the collection point of about 6 inches of pea stone. And then we come up with the 5 feet of sand, the 1 foot of trench, and then the mandatory 9 inches of cover. So within those 5,000-gallon tank shim sets, there's a bottom last that we have lined with our liner and a pipe to perform a collection procedure. And then that brings up to the top. And within that 5,000-gallon tank shim, there are 5 separate cells. The 4,000 or 5,000-gallon tank bottoms, tank shims, actually have a length of about 17 feet and a width of about 8 feet. So our cells could fit trenches that are 5 feet long, 1 foot deep, and 1 foot wide. So you can imagine behind me, you'll see all 35 of those cells with various pipes coming up, with the gravity pipes having 2 main pipes showing, one which is a clean out for the underdrain, which was thought by Dr. Pancorbo to be at least necessary to occasionally flush out so that there'd be no excess growth in there. And the other one is actually an observation port, much like our Title V observation ports, so we can observe when ponding starts to take place. Because as we all know, or at least we all believe from the common wisdom, is that once a biomat forms on the bottom of the system, it will perform a little bit differently than initially thought. Initially, what happens is where the effluent just goes down in one spot, it's evened out as the biomat accumulates. So that's pretty much it. There's 35 cells, everyone to a discharge point. The deepest ones we have going to this large structure right here, you'll see behind me. And those 15 pipes all come to one collection point where we'll have to actually situate our collection baskets or buckets for 2 liters. We need quite a bit of collection for the virus collection because we have to concentrate those. Due to the concentration of the virus or the density of the virus in wastewater, we have to actually collect 2 liters and then filter it down or run it through a filter, which is sort of akin to really what our kidney does, and then rinse off those viruses into the sample that you've probably already heard from our microbiologist. So the construction of the cell is pretty much, there's a cell, there's 5 of them within it, right? And within the cell there are 5 separate discrete areas where we can separate out the leachate from the trenches. And as we collect down at the bottom of those, there are 5 discrete samples. So in essence, these are a 5-set replicate. For statistical analysis, we definitely need that. So as you look at the diagram here, you can see at the bottom of the cell, there's a drainage area. That has got pea stone around it so that all of the water percolating down through the system after it departs the stone trench will collect down there, and the liner is sloped so that all the drainage occurs rather quickly so that it doesn't accumulate or have any biogrowth down at that point, and drains to our collection point. As we come up, you either have the 2-foot, the 3-foot, or the 5-foot of sand. And those are the real test treatment cells that we're trying to figure out how much sand is really necessary for the removal of the viruses. So then you come to the sand trench interface where the stone is actually setting right down on the sand. That's again something that's 5 feet long, 1 foot wide, and 1 foot high. And across the top of that is set your perforated pipe, your perforated 4-inch pipe, with a little blocking layer of fabric so that the silts and fines don't wash from the top down into it. Then on top of that, a tad bit of sand to hold that filter fabric down, and then from there on we have the fill, which in this area we'll be filling with like a sandy loam or a loamy sand, because that's what's typically used as fill. There'll be some organic component to it because we would like to see a nice grass on top of it so that at some point we can also start to calculate what the impact of evapotranspiration from, say, a lawn would be. Now the pressure is a little bit different. With the pressure, we have pressurized pipe sitting in that same situation as we have your 4-inch perforated pipe. And it has a clean out at the end so that we can occasionally check the residual head pressure and make sure it meets the code. So the actual construction of the cells takes place, it's a little bit complex. We had to sort of invent a form that we could actually parse the 5 cells, so to make each individual cell, put a liner in it, but we had to remove that particular form 6 inches at a time as we brought this up. Otherwise, you can't just bury the form and pull the forms out because there's too much side pressure. So what we came to is about every 12 inches. Every 6 inches we walked it in. Every 12 inches we actually raised the shell of the form and kept going until we got right up near the top, then marked it, covered it, and waited for the construction of the actual trenches themselves. The trenches themselves, we have 5 different collection boxes that we laid right beside one another, 5 containment boxes for the stone trenches, because anybody knows you can't just dig a hole in sand and hope that you'll get a nice square trench. But we're able to do this by the use of forms. Okay, this is a typical of our pressure dose trenches. This small pipe, of course, you might recognize, it's usually below grade, and it will be close to below grade by the time we're done. This is for the actual checking of the distal lateral head. And we check these all, and they have easily 2.5 to 4 feet of distal lateral head. This pipe here is for our actual ponding measurement. So when the trench starts to pond, we'll see it here, since this is the distal end, proximal end up that side. This other pipe here you won't see in anybody's backyard. This is so that the underdrain that is collecting the sample beneath our system, which in this case is 4 feet down, this can be flushed. So every couple of weeks we'll come in high pressure, just push the water through here to our collection point, make some observations about the growth. If it doesn't grow a whole lot down there, we'll probably clean them less frequently. But otherwise, it just provides us an idea of whether we're getting biogrowth within our collection device, because anytime you have biogrowth, that's going to skew your results. It's going to make it particularly bacterial. They'll grow like crazy, and they may even harbor some of the pathogenic bacteria or the pathogen surrogates that we're measuring.
We had to find a way to dose each of the buckets accurately and precisely. Of course, accurate being delivering a dose that's within about 1% or 3% of a target volume. And then precise being giving that dose a number of times during the course of the day. So what we came up with is a bucket system, which you can kind of see right here beside me, that contains an overflow inside, so that when the bucket is filled with wastewater, the barrel can only fill to a certain volume each time. It allows us to get that accuracy and precision. In order to make sure that, the number of doses actually happen during the course of the day, there are floats inside each bucket that are set about the height of the overflow. So when the bucket fills, the float triggers a counter, and then we can tell that the bucket filled to that volume, and also that the dose actually went out to the system. So after the buckets are filled and the volumes are sort of stabilized inside, is when, depending on the type of cell we're dosing, we would trigger either a valve underneath the bucket, which is an actuated 1-inch ball valve, or there are pumps inside of each of the pressure distribution systems that would pump the effluent out of the bucket and out to the mini leach field test cell. The pumps themselves are what are called skimmer pumps, so they're able to draw the bucket down to about 1/8 of an inch or so. We put together a custom-built programmable logic controller system that would monitor all the floats, turn on all the pumps, turn all the valves, and we're able to do that on a schedule so that during the course of the day we can do 5 doses. Those doses go out according to roughly an NSF, which is National Sanitation Foundation schedule, which is 35% of the doses in the morning, 25% in the afternoon, and then 40% in the evening. So for this particular project, in order to maintain a dose size that was big enough for the pumps to be able to put out enough head to each of the test cells, we chose a 2.5 gallon dose, which would require, per the size of the fields, that we do 5 doses per day. So we split that up. We'll do 2 in the morning, 1 in the afternoon, and then 2 in the evening dose period. But we do have the ability to really fine tune how that schedule works. If we want to experiment with some different loading regimes, we do have the ability to change the schedule in the system pretty easily. And we can also change the overflows in each of the buckets so that we could do different dose volumes fairly easily. So again, we'd be doing 5 doses a day, 2 in the morning, 1 in the afternoon, and 2 in the evening at 2.5 gallons. So ideally, this system will be able to function for a long period of time and give us a tremendous amount of flexibility with this project, but also really go the long haul and provide us with the accurate and precise doses that we need.
Okay, so now that you've found out all about the project that we're about to do, I now have to introduce just a little bit of metadata, not to be confused with Metamucil, and that reference is probably lost on anybody under 65. So metadata is the data or the information that informs us about our study. And the world of viruses is extremely complex and extremely diverse, just like the animal kingdom. There are all sorts of variables that affect how one goes one way or the other, how it's retained. But there are general principles that we all should know because they'll help us in our design of septic systems to sort of minimize the threat of viruses getting to groundwater. And the first one is the first factor and the first information that comes out is that about hydraulic loading rate. As with many contaminants, hydraulic loading rate is like the key factor. The hydraulic loading rate is the rate at which we apply to the soil or to a treatment media a contaminated source, a contaminated water. So in reference to what we're doing, in septic systems there is what we call a long-term acceptance rate or the hydraulic loading rate. But even there we have to parse that out. The real hydraulic loading rate, not the one you theorize or calculate with your area. In, for instance, gravity distribution, and you can see from this illustration here, for a long time the effluent really is loaded heavily on the one area where it enters. No matter how level we lay the pipe, and by the way, Massachusetts, we don't lay it level. We tilt it to a certain degree so that the effluent all goes to sort of one spot at once, and that introduces a very high instantaneous loading rate compared to this other part, which is pressure distribution, which more of the surfaces at one time have the effluent applied to them. So they expose more surface area for the effluent to actually receive treatment by the bacteria that are growing on it, as well as the nutrients that are available. So these two modes are probably the most key in virus removal. Here I show an illustration of something that was done back in 1987 by Marilyn Yates, and it's a graph that shows hydraulic loading rate in relation to virus removal. And you can see here that the higher hydraulic loading rates mean more viruses get to the groundwater, which makes sense. I think if you've seen my other sort of talks about this, this would be like dumping 1 or 2 BBs onto large columns of 12-foot boulders versus a dump truck full of them. So hydraulic loading rate is the key. The second piece of this, as you can see in this graph here, is pressure distribution versus gravity distribution. Here I've done one little study with bacteria, and it's the log removal of bacteria in relation to the distance to groundwater, the breakthrough to groundwater. And you can see from this little red star on the graph where pressure distribution is, we've achieved like a 2 log increase or 99.999% removal of bacteria just by changing the application mode. And it's not just the application mode we're changing there, we're changing the actual hydraulic loading rate. When we apply pressure distribution, we're using far more right away, initially, from day one, more of the soil. We've applied a lower loading rate and hence have achieved better treatment. The second factor would be temperature. Colder temperatures, as you may know, because we put things in our freezer or our refrigerator to slow down growth, lower temperatures also cause a higher persistence of viruses. So the lower temperatures, for instance, in the winter when our groundwater may stay the same, but our effluent and in our septic leach fields, the temperature can drop as low as 5 degrees. This would really help viruses persist. So we might see, and we'll see probably in this study, that there's a seasonal component to the virus removal in groundwater, in septic systems or beneath septic systems. The microbial activity. If there is a high microbial diversity in the soils that we're applying the wastewater to, that is generally, and I have to say generally on all of these because, again, the viral world is pretty diverse, you have a higher rate of decay of viruses if there are a lot of bacteria looking around and scavenging for nutrients, because basically a virus is an inert particle, has some nutrients in it, has phosphorus, has nitrogen, has all the DNA and RNA components. So eventually, as those break down, or there's bacteria that have lytic compounds that can break them down, a high diversity of bacteria is usually, as with any other contaminant, is usually a little bit better at removing it. Moisture content of the soil. That's another key factor. The distance off groundwater controls the moisture. If you're very close to groundwater, and well, you'd be able to see this. Put a pile of sand out in your backyard, and when a rain comes, and after it's done, you'll see that there's a capillary area right beneath the bottom of the sand that comes up that indicates how high the moisture comes up. If you have a clay pile or a pile of clay and silt, that moisture can actually be capillaried up a lot higher. The higher the moisture content in the soil, the more the virus can survive. And ultimately, if it gets to saturated conditions like groundwater, there's basically nothing that will stop its movement except long-term decay rates. Eventually, everything breaks down, and at 68, I'm a testimony to that. pH. pH is another key factor. pH controls really whether a virus lets go of something or whether it hangs on to it, because pH modifies the charge on the outside of the particle. All clays and some soils and mineral soils have a charge to them. A lot of the clays are negatively charged. A lot of the viruses, under low pH conditions, that little proton hangs on to the outer coat of the virus capsid and makes it charge, and it will hang on to the soil. It'll hang on to a clay particle. So lower pHs generally are conducive to more virus transport, more virus hanging on and persisting. So this again is a general principle. Some viruses don't have that charge. They have different points which they let go. By the way, that's called the isoelectric point. The pH at which everything is neutral and that virus then can move toward the groundwater. The final part, the soil properties and everything, relates to organic matter. If there's a lot of organic matter, that throws all the equations off. Organic matter has charge, so it attracts things both in one or two different ways. And so, in general, what the metadata show us are that soil properties and the properties of the wastewater do a lot of driving of whether a virus persists. So that's it on metadata, and we'll close it out with some final remarks.
At this point, we've finished the dosing. The cells are all constructed. We've got the sampling pretty much set up, and we should be able to start actual analyses within the next few weeks. So by the time you're watching this, we should have some data available, and we're really excited to kind of get this project going.